Hi everyone, welcome back to our molecular biology series. Today we are solving a fascinating puzzle. How does a human cell fit 2 meters of DNA into a nucleus that's only 10 micrometers across? That's like fitting a rope that's a 200 kilometers long into a room the size of a basketball. The answer is packaging. Elegant, efficient DNA packaging. Let's see how cells do this incredible feat. Let's understand the scale of this problem. A human cell has about 6.6 billion base pairs of DNA. Each base pair is separated by 0.34 nanometers. So, if we multiply total base pairs by the distance between them, we'll get approximately 2.2 meters of DNA. That is taller than most people. But the nucleus where this DNA lives is only about 10 micrometers in diameter. That is 0.00001 meters. We cannot even see it without a microscope. Imagine trying to fit a 2-meter rope into a space smaller than the full stop at the end of a sentence. Impossible, right? Yet every single cell in our body does this. The solution is packaging, brilliant and organized DNA packaging. Let's start simple. How do bacteria like E. coli package their DNA? They don't even have a nucleus. But their DNA isn't just floating around randomly. It is organized in a region called the nucleoid. Here is the trick. DNA is negatively charged because all of those phosphate groups in the DNA backbone, if you remember from our video about DNA structure, the cell uses positively charged proteins to grab onto the DNA and organize it into large loops, like gathering a long rope into neat coils. Opposite charges attract, so the negative DNA sticks to the positive proteins. This keeps the DNA organized even without a membrane bound nucleus. Eukaryotes, on the other hand, like plants, humans, or fungi, we have way more DNA than bacteria. So we need a more sophisticated packaging system. Then comes the histone. Histones are special proteins that are positively charged. Why? Because they are packed with two amino acids, lysine and arginine, and both of these amino acids they have positive side chains, positively charged side chains. And because opposite charges attract, the negatively charged DNA wraps around these positively charged histone proteins like a thread around a spool. Each packaging unit is made up of 8 histone molecules called a histone octamer. Octa means 8. So these 8 histones work together as a team to package DNA efficiently. When DNA wraps around a histone octamer, it forms a structure called a nucleosome. Think of it like a thread wrapped around a spool. The histone octamer is the spool and the DNA is the thread wrapping around it. Each nucleosome packages about 200 base pairs of DNA. The DNA makes about 1.65 turns around the histone core, not quite two full circles. Now, here is a mind blowing number. If a human cell has 6.6 billion base pairs and each nucleosome packages 200 base pairs, that means we have approximately 33 million nucleosomes in every single cell. 33 million little spools of DNA all neatly organized. Nucleosomes are the basic repeating units of chromatin, and chromatin is what we call this packaged DNA protein complex in the nucleus. Let's try to draw a nucleosome to see how DNA is wrapped around it. So these are our histone proteins. This is a nucleosome and I will color code it. So these two circles are H2A, these two circles are H2B, and these two circles are H3 and H4. This is histone octamer, and here is H1 or linker histone. Now this is our DNA wrapped around it. So this blue thread is our DNA wrapped around nucleosome. So here is the histone octamer, 8 histone proteins clustered together forming the core. So this one is the histone core. 2 copies of H2A, H2B, H3 and H4, and there is also H1 or linker histone. Now the negatively charged DNA wraps around this positively charged core, making about 1.65 turns, and this is not two full turns. This is somewhere between 1 turn and 2 turns. So and this 1.65 turns contains around 200 base pairs of DNA. The DNA enters on one side and exits on the other. So this is just one nucleosome, and like this there are multiple nucleosomes. This is other, and then this is the third nucleosome. So multiple nucleosomes strung together to create the beads on string structure. Each bead is a nucleosome. This is nucleosome 3, this is nucleosome 2. So the string is the linker DNA. So the DNA will come like this. So this DNA that bridges or that connects two nucleosomes together is linker DNA. This is the first level of packaging. Now the key parts to remember is the histone core, the DNA wrapping around it, the linker DNA between two nucleosomes. I am using this yellow color. So this is our linker DNA, or this is our linker DNA. This structure reduces DNA length significantly and also helps regulate which genes can be accessed. When you look at chromatin under an electron microscope, nucleosomes appears as beads strung along a DNA thread, literally called the beads on string structure. Each bead is one nucleosome, which is DNA wrapped around histones. The string connecting the beads is the linker DNA between nucleosomes. This structure is beautiful and functional, but this is just the first level of packaging. The DNA is compressed a bit, but we need to condense it much more to fit everything into the nucleus. The beads on string structure coils and folds further into thicker chromatin fibers. These fibers coil again and condense even more. This higher level packaging requires additional proteins called non-histone chromosomal proteins or NHC proteins. These aren't histone, but they help organize and compact the chromatin even further. The ultimate condensation happens during cell division at metaphase, when chromatin condenses into the familiar X shaped chromosomes which we have seen in our textbooks. That's when DNA is maximally compacted and ready to be separated into daughter cells. Not all chromatin in the nucleus is packaged the same way. There are two types. Euchromatin is loosely packed chromatin that stains light when we look at it under a microscope. U means true or good. This is active chromatin where genes are being transcribed. The DNA needs to be accessible for RNA polymerase to read it, so it can't be super tightly packed. Heterochromatin, on the other hand, is the opposite. Hetero means different. It's very tightly packed, densely compacted, and it stains dark. It is transcriptionally inactive chromatin. Genes in this area are essentially shut off. The tight packaging prevents access, so transcription cannot happen. Think of euchromatin as open books you are reading and heterochromatin as books sealed shut on a shelf. Both are in your library, but only one is accessible at any moment. This is actually a form of gene regulation. Packaging controls which gene can be expressed. So, the summary is to fit 2.2 meters of DNA into a minute nucleus, cells use stratified packaging, which is multiple levels of compaction. In eukaryotes, DNA wraps around positive histones to form nucleosomes, the basic beads on a string unit. These coils into fibers and eventually condense into chromosomes with the help of non histone proteins. The structure also regulates gene expression. Loose euchromatin is active while dense heterochromatin is silenced. Essentially, packaging solves the space problem while controlling genetic access. For NEAT, remember these key points. If this video helped you understand DNA packaging, please follow for more such videos. Thank you for watching.